as an older woman, we should not get pregnant anymore. There's no question whatsoever if men should get pregnant or have kids, sorry, at 45 years old. They might not be as active anymore as you see them here, but nobody really doubts that there could be any problems. Yet, most of these genetic conditions that you see here all come from older men. In fact, the majority of the genetic conditions come from older men. And this has been the topic of my research since I've been a PhD. Why do older men have so many mutations that are passed on to our offspring? Changes are particularly dangerous if they occur in the germline, meaning the person is normal, but they produce mutant or a mutant sperm or mutant eggs. And when this happens and this mutant sperm fertilizes the egg, it can be that entire organism or children are affected and carry the mutation. In this case, for example, achondroplasia. Fortunately, most of the new mutations in the germline are very rare. In fact, they are 100-fold rarer than somatic mutations in our tissue. And however, there are still 30 mutations per genome per generation. But this doesn't explain why older men have more mutations. So I first want to tell you a little bit about sperm and egg formation. So on the left hand, you see the male germline formation compared to the female formation of eggs. And the take-home message is that most of the divisions where you replicate and copy the DNA happen during embryogenesis, and in the female, you stop with the copying even before reaching puberty. Versus in the males, you keep copying and dividing 23 times a year these spermatogonial stem cells to produce the sperm. And this happens throughout the life of the male. So if you then, for example, count how many cell divisions happened at age 20, this is almost 8 times more than in the female. And even with age, you can see that we have 4 times more cell divisions with as many mutations in the male germline. So basically, if you think about copying being error-prone, which actually any copying procedure is, it might explain why you accumulate with age more mutations. But there's another thing that is important here, way more important than this copy-error hypothesis. And that is, we're talking about stem cells. Males have stem cells in their germline. Why is this important? There's this whole theory about stem cells and selfish mutations. What are these selfish mutations? Well, if you look at cancer, basically tumors start growing because stem cells get a mutation, and this mutation changes their dividing behavior. So now they start behaving more often, more frequently, and start forming these tumors or clusters. So these selfish mutations or driver mutations, they promote their self-propagation. They also only act in dividing or replicating tissues, that is, you need stem cells. And this is exactly what you also have in a germline. These mutations happen in the DNA, and there's a way of measuring this. So my research focus has been trying to identify these driver mutations in the male germline by looking at the DNA. And this is what this LIT project is about. I've selected a gene that has accumulated a lot of mutations, at least in tumors. It's actually a growth factor, FGFR3 short. And if I look at the tumor databases, I found at least 277 changes in this gene that have been found in tumors. Now that's quite a bit for a gene, actually. Comparatively, these are the mutations that have been associated with congenital disorders, the ones that I've been talking about at the very beginning, meaning that they have been associated with a genetic disorder. Only 35. Now the question is, why do we have such a large difference between mutations happening in this exact same gene in tumors compared to germline? Is it because somehow the germline is protected, or is it because we just haven't found them and nobody has measured these driver mutations in the germline? So These are known because a congenital disorder was described and it was linked back to this gene. So in our LIT project, we want to actually now discover these mutations by sequencing. How do we do this? Well, we take sperm DNA, extract their DNA, and try to find this rare mutation. Now, that's easier said than done. The problem is finding these rare mutations. It's like searching a needle in a haystack. So you have many wild type or non-mutants, and you try to find out one single nucleotide change within all these millions of bases. So our approach is maybe not finding the needle, but it's more like counting the needle. Ultimately, we don't care where it is, we just want to know how many needles the haystack has. So our approach is to basically turn out and look at every single piece of hay or DNA and ask, are you a mutant or are you a wild type? And then ultimately, doing this in parallel, you can look at millions of DNA molecules at the same time. Now, another problem when you try to find a rare event is trying to make sure that this is not a technical artifact. So you want to make sure that you're measuring something real and not a technical error. So for this purpose, we have come up with a creative molecular design in order to distinguish real from artifact. And the first thing what we do is to cut out a piece of our genome, in this case a small piece of our FGFR3 gene, that we then sequence millions of times. So we really make many, many copies that we look at. During this copying process, so these are the pieces that we cut out here in yellow, we actually tag them. And for this tagging strategy, we use 4 to the power of 12 different tags. And then we tag them left and right, and then we create multiple copies of each and every single molecule. And we create not only the copy of one strand, but in fact we have copies of both strands. If you remember from your high school classes, the DNA is formed by 2 strands, the forward and the reverse strand. And we actually do have copies from both of them, and we can track them back. Why is this important? Well, now we can distinguish errors from true mutations the following way. So let's assume that we have occasionally a sequencing error or some kind of artifact error. Well, it's highly unlikely that that error comes up every time at the same place in the copy. So by building these families of copies, you can see, well, most of them don't have the error, so this is probably an artifact. Now, you can be unlucky and see that the error occurs very early and starts propagating at the same place. That's why it's important to have the sequence of the reverse strand, for example, and check, well, does it occur there too? And if it doesn't, then you know, okay, it's also an artifact. So only true mutations that happen in the forward and in the reverse strand are going to be considered as true mutations. And with this, actually, we have one of the methods with one of the highest sensitivities out there that can be used to discover these rare mutations. We still have the problem that we have to create a lot of data to only have a small glimpse of what's happening. So we create about 7 million sequences in one example. Then we create these families with 8 to 18 members per family. The ideal scenario is only to have 4 or 5. And even worse, we're losing even more data when building these duplex consensus sequences where we take together the forward and the reverse information, where ideally you'll have 2 single-stranded consensus for 1 duplex consensus. So right now we're still needing, for analyzing one molecule, 180 copies, which is quite a bit. And ideally we want to get down to 8 to 10 sequences. So what we're trying also to develop is some smart bioinformatic analysis and trying to figure out where we're losing data. This is one example where that shows you in the tags of in all the different tags that are being used. In theory, we should only have differences of 5 to 6 nucleotides between tags. And in fact, we see a lot of tags that only have 1 difference. So it's also very likely that these tags have a sequencing mistake. And by allowing a mismatch, for example, in the tag, you can reallocate the data and salvage data that was misplaced in the wrong families. So these are kind of the things that we're doing. Now I show you a little bit of real data. This is the region you're sequencing, if the FGFR3. This is some of the data. These are the number of mutations. These are the number of molecules we looked at, the mutation rate, and the type of mutations we see. The nice thing about it is that we're picking up mutations that have been already known to be quite frequent, causing this congenital disorder. In a second data set, we're also seeing similar mutations, so we can reproducibly pick up the same mutations. Now, the scary thing about this whole data set is, if you remember, the mutation frequencies in the germline was 10 to the minus 8. Maybe I cannot do math, but these are 4 orders to 5 orders of magnitude higher. I was really scared when I was looking at this data because this means that our conception of how frequent mutations are in the germline is completely wrong, and we're having events that are way more frequent than we have ever thought before. All right, so we're starting to get data that show that these selfish mutations are active in the male germline, and we can start measuring them. We are now developing more data sets and to see how they're expanding, whether truly expanding with age, if they're expanding also in sperm, and maybe you can take home this message that you want to, as a young student, you might form a family sooner than later instead of building up a career. But this is, of course, everybody's choice. But thank you for your attention.